Okay, it looks like we are on air. Thanks everyone for joining in for today's presentation. Just give a quick introduction to myself. My name is Wes Chang. I'm a senior staff scientist in the Cell Analysis Business Unit, the former molecular probe site in Eugene, Oregon, now part of Thermo Fisher Scientific. I'm actually relatively new to Thermo Fisher. I've been here a little over 6 months. Prior to that, I spent about 12 plus years working in the biotech pharma space in the Bay Area in California. Have experience working at companies like Genentech and Amgen, and during that experience I was mostly focused on cancer therapeutic development and first sort of got some grounding in hypoxia research when I was studying VEGF signaling at Genentech. But that being said, I've also participated in projects in the metabolic and cardiovascular space, as well as neuroscience, immunology, and inflammation. My training is actually in neuroscience and developmental biology, but I consider myself to be more broadly a cell biologist with lots of experience in microscopy and emerging experience in flow cytometry, focused on biomarker assay development and immunohistochemistry and pathology. So, the sun has not come up here in Eugene, so but thank you all for joining at this early hour, at least here on the West Coast. There's an outline for today's presentation, and I would say generally it's meant to be a largely educational presentation. We're trying to target a broad audience, and it's not necessarily meant to be a deep dive into any specific area of hypoxia research, and because we want to cover a lot of ground, we're going to sacrifice depth in any one given area. But really the goal is to introduce key concepts in oxygen sensing, cellular responses to hypoxia signaling, and talk about some of the experimental approaches and methods that are used to study hypoxia. We're going to talk about examples of disease pathologies where hypoxia is a contributing factor. And once again, we'll talk a little bit about how hypoxia has traditionally been studied in the laboratory, but then also introduce a couple of newer reagents and tools that can be used that are more recently developed. And hopefully by the end of the presentation, if I'm successful, you'll come away with new ideas and insights into how hypoxia may be relevant to your own research if you're not already deeply involved with it. Now, for logistics, because of the webinar format, we're going to reserve time at the end for question and answers. So I believe viewers can enter the questions into the Q&A window at the bottom of the screen, and at the end of the presentation we'll try to get to as many of those as we can. So just as a starter, I think it's worth, maybe at the risk of stating the obvious, that oxygen is really important for most life processes on Earth. It's a selective and evolutionary pressure for somewhere on the order of 500 million years on the planet. It's worth pointing out that for the first 3 billion years or so of the Earth's history, there really wasn't much oxygen in the atmosphere, and it wasn't until the evolution of photosynthesis, and in particular vascular plants, where the atmospheric oxygen levels rose dramatically, and that sort of prompted the advent of oxidative phosphorylation, which was, of course, a key driver in the evolution of complex metazoan life forms. So in order for this to happen, we had to develop very sophisticated systems for delivering oxygen to cells, such as the circulatory system and the respiratory system, and as well as methods to monitor oxygen levels and produce adaptive responses too low oxygen levels. I also just want to point out that this curve is presumably not totally correct in that one would think that it would level off here. It's not going to keep going up. But you can see that life on Earth really took off when the oxygen levels got high enough to support metazoan life. So I think this slide's kind of worth spending a couple minutes on in terms of thinking about the differences between Normoxia and what some people like to call Physoxia, and contrast that to hypoxia. So atmospheric oxygen is present at roughly 21%, of course, depending on what your elevation is and whether there's a lot of air pollution, those kinds of factors. But that's quite a bit higher than the actual concentration of oxygen within your tissues inside your body. And I think, that is generally thought to be in the neighborhood of 5 to 12%, roughly, but that is also very highly tissue dependent. So I think it might be most instructive to think about hypoxia as a concentration of oxygen where cells and tissues begin to show adaptive responses in response to limited oxygen availability. And in many cases, we're talking about the situation where the oxygen concentration falls below 5%, but once again, highly cell and tissue dependent. And so I think what's important is that especially if you're thinking about in vitro experiments where you have cells that are growing in the cell culture incubator in 2D culture, generally speaking, the oxygen concentration is the same as environmental air, although there is also 5% CO2 typically being added into the mix. So the oxygen concentration inside of your standard cell culture incubator is roughly about 20%. And once again, it's not exactly clear how much oxygen the cells in 2D culture actually experience. It's somewhat dependent on how much cell culture media you have in your buffering system. But I think it's generally a safe assumption to say that what cells see in cell culture is probably a bit higher than what tissues are actually seeing in vivo. Now, the lower chart below is actually quantification of oxygen percentage in normal tissue shown in green. So once again, this is sort of where I'm drawing this average of about, a little over 5% normal oxygen in normal tissues compared to the oxygen concentration that's been measured with oxygen probes in a variety of solid tumor indications. And the general theme is that in cancer, in solid tumors, you have hypoxia, and that's been known for a long time. And actually, the adaptive responses within solid tumors can actually have adverse consequences. And we'll discuss these concepts in a couple more slides when we talk about disease pathologies. So what kind of adaptive responses are we talking about? And to get into that, we have to sort of introduce the very well characterized HIF-1 alpha pathway, which is the molecular mechanism for oxygen sensing and signaling. Now, the key players in this network are the hypoxia-inducible factors, which are or also known as HIFs, and these are a subfamily of basic helix-loop helix transcription factors that were initially discovered and described by Semenza's lab at Johns Hopkins. And I'm going to go through this really briefly because I think the pathway is sufficiently complex, and I think it's pretty well appreciated for anyone who studies hypoxia. In Normoxia, under normal oxygen conditions, HIF-alpha is constitutively hydroxylated by the activity of enzymes called prolyl hydroxylases. So these PhD 1, 2, and 3 enzymes are responsible for keeping hydroxyl groups on HIF-alpha. And what that does is it creates a binding site for the von Hippel-Lindau gene product, which targets HIF-alpha for degradation in the ubiquitin proteasome degradation pathway. So what this means is under conditions of normal oxygen, HIF-alpha is tightly regulated and generally kept quite low. And also, under normal conditions, HIF-alpha is maintained in the cytoplasm. So, however, under conditions of hypoxia, then this hydroxylation does not occur. So von Hippel-Lindau does not recognize HIF-alpha, and therefore it's not ubiquitinated, it's not targeted for degradation, and instead HIF-alpha can enter the nucleus where, with assistance from CBP, Krebs-binding protein, also known as P300, it can dimerize with HIF-1 beta, and that will form a functional transcriptional unit which is able to recognize and bind to hypoxia response elements, which are located upstream of hypoxia-inducible genes. Like any good pathway that's very tightly regulated, there's also another mechanism for making sure that HIF levels are kept under control and in the right place, and that's this molecule called factor inhibiting HIF, or FIH, and that is actually another kind of enzyme that adds hydroxyl groups to asparagine residues. So, as you can see, there's multiple levels of regulation, and that's just the hallmark of any complex, important pathway in biology, in my estimation. Now, like any good, complex pathway, there's a number of points for pharmacological intervention where drugs can be added to inhibit or activate different steps along the pathway. So in red here, I've just shown a couple of the most commonly used compounds that are used to perturb the system. So there's compounds that will sort of inhibit the activity of the PHD prolyl hydroxylases, including DMOG and DFX. DMOG is an alpha-ketoglutarate analog, which can inhibits the enzyme through that sort of mechanism, whereas DFX is an iron chelator, so the PHD enzymes are iron-dependent. So those are a couple different places where you can affect prolyl hydroxylase activity. There's also classical compounds like digoxin, cobalt chloride, which is one of the most widely used compounds to study hypoxia in cells, acroflavine, which inhibits the dimerization of HIF-1 alpha and beta, and then newer classes of compounds which are more specifically targeted against ubiquitination and proteasome functions, such as this millennium compound here. So, like any of these compounds, and I think you get a sense from the description of how these things work, that none of them are truly specific for hypoxia, and that all of them have some associated toxicities. So, these can be thought of as hypoxia mimetics in that they induce a hypoxia-like state in cells by affecting one portion of the pathway or another, and but there's really not a substitute for actually reduced oxygen concentration and understanding what happens to cells. But nevertheless, these tools have been able to provide some useful mechanistic insights into the organization of the pathway. It's worth pointing out that pharmaceutical industry and biotech is developing newer compounds to more specifically target points in the pathway. And there's actually quite a bit of emerging clinical data on these molecules, but it's really beyond the scope of the presentation today. And I would, point you to some of the excellent review in the literature that cover those topics specifically in terms of hypoxia-targeted drug development. So what are these genes that the HIFs can regulate? And depending on, the methods you use and the literature you look at, there's upwards of 1,000 genes in the human genome that can be regulated by HIFs. And although it's worth pointing out that only a subset of those will be activated in any given cell type under hypoxic conditions. So two themes that emerge in terms of looking at the targets are you see genes that are involved in increasing oxygen delivery to cells and tissue, and perhaps the best known examples are things like erythropoietin. I mean, the erythropoietin was central to the discovery of the hips, and then of course genes that increase the blood vessel formation, angiogenesis. So all the proangiogenic factors are known to be regulated by HIF. And then another class of genes are genes that are turned on to decrease oxygen consumption and basically push cells towards a glycolytic metabolism. So you see that sort of pairing where the cell on the one hand is trying to conserve its energy, its oxygen consumption by switching to glycolytic metabolism, but at the same time is trying to compensate for reduced oxygen by increasing the very mechanisms that deliver oxygen to cells and tissue. This is by no means meant to be a comprehensive list. I just wanted to show that there are many, many different cellular functions that are enhanced or affected by hypoxia, including genes that are involved in cell motility, and I would say that it's one of those cases where you can choose your favorite pathway, choose your favorite gene or cellular function, and there's a good chance that it's probably influenced by oxygen and or hypoxia. So at the level of individual cells, a variety of responses to hypoxic environments have been very well documented, and this slide is meant to capture that in sort of a simplified cartoon fashion. Now, and we've broken it down into thinking about acute responses versus chronic responses, and acute responses are things that are occurring on time frame of seconds to minutes following oxygen withdrawal. And then more chronic responses, which take days and can last for weeks, and those are ones that generally involve changes in gene expression. So one of the most fundamental changes that occurs acutely in response to low oxygen is a decrease in the ATP to ADP ratio. And that's largely due to impaired oxidative phosphorylation, although there's other mechanisms that also decrease this ratio. So because ATP is synonymous with cellular energy, you get all the things that ATP is required for are therefore also affected. And one of the most profound targets is the maintenance of ionic gradients, and here just the example of the sodium-potassium ATPase, and these are required for maintaining ionic gradients and electropotential across cell membranes. So a very large amount of cellular energy generated in the body is actually geared towards maintaining those ionic balances across membranes. So once this ratio goes down, you start to have problems with ionic balance, and that can affect many, many different aspects of cellular function and signaling. Some of the things that happen, as I mentioned, you switch over to a glycolytic metabolic pathway, so you get an increase in lactic acid in cells and tissues, increased carbon dioxide, and this causes a drop in extracellular pH. For people who are used to thinking about exercise, they know that this is why you get sore after you work out, and it's basically it's a conditioning sort of thing where the skeletal muscle, which is actually pretty resistant to low oxygen, can actually get conditioned and build strength after this sort of acute damage to the tissues. There's a controversial role of what's going on with reactive oxygen species, but there's evidence that hypoxia does lead to an increase in ROS production from mitochondria. Not surprisingly, very complicated pathways such as DNA repair, which I'm sure most people are aware was the subject of the 2015 Nobel Prize in Medicine. Those DNA repair is ongoing all the time, and it's, lots and lots of enzymes are required to maintain, proper genome stability. And so under conditions of hypoxia, those mechanisms start to fail or start to exhibit errors, and what that leads to is increases in genomic stability. And we'll talk a little bit more about that when we get back into oncology. And similarly, all the things that make your proteins fold properly and transit through the secretory pathway can also be effective, and you get sort of increase in ER stress related to the unfolded protein response. And then in the more chronic sort of examples, as I mentioned before, you get transcriptional activation of proangiogenic factors and receptors in some cases that mediate the remodeling of blood vessels as the body tries to deliver more oxygen to cells and tissues that need it. And in the worst case scenario, where all these things kind of come to play under chronic conditions, you can cause the cells to die. Okay, so now we're going to transition to discussion of how hypoxia interacts with various different disease states. And we'll start with talking about the cardiovascular system. And, given that the cardiovascular system is evolved as a mechanism to deliver oxygen throughout the body via hemoglobin and red blood cells, it's not surprised that these diseases that affect the circulatory system intimately involve hypoxia. So ischemia, which is basically the inadequate tissue perfusion, delivery of oxygen, as well as reduced clearance of toxic metabolites, is a hallmark of many cardiovascular diseases. And here on the right is sort of a little cartoon describing once again that hypoxia as a central organizing principle. Your body's response is to try to increase oxygen supply and delivery while at the same time decreasing oxygen demand. And what ends up happening is that you get activation of proteases. As I mentioned before, you get changes in metabolism, increased glycolysis, decreased pH. You get increased in angiogenic signaling. You also get release of pro-inflammatory cytokines, and also alterations in lipid metabolism. And if you kind of put this all together in the context of coronary artery disease, which, as we know, is the leading cause of death in the U.S. population, you get something that amounts to a vicious cycle of hypoxia, necrosis, inflammation, which can lead to weakening and eventual catastrophic rupture of the arterial wall in a myocardial infarction. So, real briefly, and I apologize because this is a gross oversimplification of what happens in coronary artery disease, but it all starts with inflammation, like so many things in terms of chronic disease. There's a vascular insult, which leads to attraction of monocytes into the location in the blood vessels. The monocytes locally can differentiate into macrophages, and the macrophages, as they try to gobble up all the sort of lipid deposits and cholesterol that are locally in the area of the plaque, they actually turn into foam cells, which are a hallmark of atherosclerotic plaques. And basically, you can get, as I mentioned, increased proteolytic activity. You get weakening of the arterial wall, and what can happen is this plaque will get bigger, and you get further occlusion of the blood vessel, and eventually, in the worst case scenario, the plaque will actually rupture, and that's really what leads to catastrophic events such as myocardial infarction or heart attack or stroke, depending on where it happens. So as I mentioned, there's effects on the immune system and inflammatory cells, very complicated regulation, and in a gross oversimplification in a way of thinking about it is that hypoxia has been shown to amplify the activity of the innate immune cells. So once again, the monocytes, the macrophages. while also simultaneously suppressing the responses of the adaptive immune system. So the adaptive immune system, I mean, if you've ever seen videos of T cells running around in the body or in cell culture, you know that they are really, really high energy consumers. So basically you can get an immunosuppression effect in a hypoxic environment where the cells can no longer do their thing. They're not as mobile and they're not able to recognize targets that they're designed to recognize. Furthermore, there's evidence that hypoxia interacts with NF-kappa B signaling and can stimulate the production of regulatory T cells and also increase extracellular adenosine, and these all, again, contribute to dampening T cell effector functions. Okay. So some of the disease states, I just want to briefly mention that I think there's a lot of interest in understanding how hypoxia interacts with inflammatory bowel disease, and there's excellent literature on this, but suffice it to say that there's a real oxygen gradient across the colonic epithelium, and that the cells that actually face the lumen of the colon are in a very hypoxic environment, and that actually becomes more so with disease. So there is a definite role for hypoxia in IBD, and because that is a big clinical need, it might be an area where hypoxia-targeted therapies could be advantageous. And then finally, in cancer, which perhaps is where hypoxia is most widely well known and understood and appreciated, you have basically what amounts to a perfect storm where hypoxia is a central player. And so all of the defects in cellular functions that we've already talked about can come into play in a larger tissue or organ level in cancer, solid tumor cancer biology. And a couple things to think about are this idea, once again, that cancer cells end up with a growth advantage over their normal neighbors, and that they become less dependent on oxygen. They can switch over to anaerobic metabolism. You get things like the Warburg effect, which really provides a survival advantage and a growth advantage to cancer cells. And as I mentioned, instability at the DNA level, which can lead to tumor heterogeneity or evolution of the tumor, and that can have consequences regarding resistance to therapeutic treatment and an increased propensity for metastasis. And actually, one of the places where hypoxia really comes into play is in mediating resistance to radiotherapy as well as chemotherapy. So for radiation treatment to work in terms of targeting tumor cells, it's really based on the localized generation of reactive oxygen species. And so if oxygen levels are depleted or low in the tissue, then the ROS species are not generated at the kinds of quantity that you need to kill the cells. In chemotherapy, there's certain chemotherapeutics that are actually prodrugs that need to be activated in the tissues, and if oxygen is low or depleted, that conversion may or may not happen, and so the chemotherapy becomes less effective. As mentioned, immunosuppression. I mean, this is a very large field for the emerging world of immunotherapy, in terms of, not only do you have to make sure your T cells and your effector cells are targeted to the tumor so that they can do their job and, through checkpoint blockade, but that in itself is not necessarily enough in that you have to overcome the immunosuppressive environment. So all these different things can factor into promoting cancer, making it worse. And one thing that I'm going to segue into here is that it's also become increasingly appreciated that hypoxia is involved in induction of not just cancer stem cells, but in stem cells in general. So, and once again, this is a huge area of biology in and of itself. I don't, really have time to delve into this very well, except to say that, stem cell biology is definitely affected by hypoxia, and that's both, the good guy stem cells as well as the bad guy stem cells. And in terms of, pluripotent stem cells that normally exist in different tissues, either mesenchymal, neuro, or hematopoietic, these all are known to exist in regions that are, hypoxic niches, basically. And it's also appreciated that maintenance of pluripotency is also requires a hypoxic environment, as if cells have to kind of be reset or shocked into some sort of altered state. And so even with respect to pluripotent cells in culture that can be reprogrammed, it looks like they have to go through a hypoxic state in order to achieve and maintain pluripotency. And then, as mentioned, in terms of cancer stem cells, these are cells that are located in these niches where basically they are resistant to treatment, and they are the cells that are responsible for minimal residual disease and relapse, recurrence. And this is actually a really huge area of therapeutic development, is trying to understand how to target the residual disease, because this is really what causes metastasis, and this is what kills people. Okay, so that's a very sort of quick tour through different disease states, and now we're going to talk a little bit about methods in the laboratory for studying hypoxia. So not surprisingly, if you want to study hypoxia, you better have really good mechanisms for controlling the environment in terms of the oxygen concentration that you're delivering to your cells. And shown here is an example of one of these traditional glove box chambers, which allow investigators to perform the entire process of their experiment in a controlled oxygen environment. Now, I think that this is probably in many ways the gold standard, because, you can have an incubator, you can do all your manipulations all under controlled oxygen concentrations, as opposed to having to move things in and out of incubators or sealed chambers. But, they're big, they're bulky, they're expensive, they're probably an ergonomic nightmare to work in, so they do have some limitations. And then also, as I mentioned, a lot of the chemical compounds, with cobalt here as an example, are useful, but they have associated toxicities, not surprisingly. So I think there's an emphasis or an effort to try to move away from these kinds of crude chemical tools and develop more sensitive, specific drugs to perturb hypoxia signaling, as well as probes for visualizing hypoxia in cells and tissue. So as far as understanding the effects of hypoxia in tissue, one of the most widely used methods revolves around these nitroimidazole probes. Now, these are compounds that get sequentially reduced in a hypoxic environment, and which allows them to form stable adducts with cellular proteins that are in the immediate vicinity of where those drugs happen to be localized. So in the case of pimenidazole, there are antibodies that will detect pimenidazole adducts in tissue. So what we're looking at here in this image is a tissue section in squamous cell carcinoma of the larynx. And what you can see here in the center is the necrotic core. In the immediate area adjacent to the necrotic core is the zone where pimenidazole has bound and is localized, detected with an antibody. And then just outside of that is immunostaining for carbonic anhydride 9, which is known to be a HIF-regulated gene. And so you can see that you get HIF expression in the region here, which leads to upregulation of HIF-regulated genes. And then finally, in gray over here is CD31 labeled vessel, just emphasizing the point that this is all happening at a distance from the blood supply. Now, some of the limitations of the nitroimidazole probes and these have been around for upwards of 20 years now, is that they're somewhat limited in terms of their sensitivity. They require very low oxygen concentrations in order to form those adducts, and so they're not particularly sensitive. And then also, because of the way they need to be delivered, they actually need to be injected into animals, or in this case, into a patient, before the tissues are resected. And they actually need to be present for a pretty long period of time, so that there needs to be a high level of exposure of the compound. And then also, to make matters worse, the binding of the nitroimidazoles is reduced in the low pH of the tumor microenvironment. So these contributing factors limit the utility and sensitivity of this approach. And then similarly, there are good antibodies that recognize HIF-1 alpha, and they can be useful for immunofluorescence detection, or IHC, in human tissues, tumors. But the main limitation here is that you're really limited to studying this phenomenology in fixed cells, post-hoc analysis, and it's not exactly the best workflow. But it's something that is definitely commonly used, and is another arrow in the quiver of the hypoxia researchers' toolkit. Now, talking about some of the newer methods that are emerging studying hypoxia and various kinds of new solutions, now we have better incubators, incubators that actually have a separate line in the back where you can feed an oxygen tank and then control the oxygen just like you would control CO2 levels. And so that's very helpful. And then, of course, they have sensors and systems for monitoring the oxygen concentration. So you can set a target oxygen concentration, and the chamber will equilibrate automatically. Now, that's opposed to these little sealed chambers where you basically gas the chamber with predefined oxygen concentration. It's crude but effective, but it doesn't allow you to have any kind of temporal control over what's going on in the chamber. It's basically just to create a very hypoxic environment, basically. And what I wanted to spend a little bit of time talking about is this fairly new product that's part of our EVOS microscope line. It's called the On-Stage Incubator, or OSI. And basically, it's an environmental chamber that's fully integrated for use with the EVOS FL Auto system. And it basically represents a very cost-effective, small footprint, robust way to control CO2 as well as oxygen, as well as temperature and humidity on the stage of the EVOS microscope. And to my knowledge, is actually one of the most cost-effective methods for doing live cell imaging. And so this is really just to show you that it's a part of an integrated system, and it really is designed to provide a complete solution for researchers who want to perform live cell time-lapse imaging studies, but maybe don't necessarily want to invest hundreds of thousands of dollars in a full-blown chamber system. So once again, the EVOS, if many of you guys have used this system before, it's actually a really nice, easy-to-use system. Everything is driven through a single software interface. There's a range of different vessel holders that are compatible with the chamber. And it's really designed to provide a simple, easy-to-use system for researchers. And this is a screenshot of the software showing the window where you would actually enable the chamber and set your target concentration of oxygen, and you get your real-time feedback over here for what's going on in the chamber, real-time monitoring. So it's a really nice sort of convenient system for doing these kinds of experiments. For anyone who's had to struggle with a multi-vendor, multi-component complicated imaging system with a big plexiglass chamber and lots of little different boxes that you have to adjust knobs on, or each one having its own separate software interface. This is actually a real bonus. It's much, much simpler to use. So also on the other side of things, there has been improvement in new probes for detecting hypoxia in living cells and living tissues. This is the IMAGE-IT hypoxia reagent from Thermo Fisher Scientific, a demonstration of what this sensor looks like in cells that are being subjected to varying concentrations of oxygen on the EVOS OSI system. And basically, you start to see fluorescence with this sensor at roughly about 5% O2. So some of the other sensors really don't show any signal until you go down to almost 1%, and by that time, it's a very hypoxic environment for most cells. And the other thing that is not shown in this particular set of images is that the fluorescence of the probe is reversible, so that if you then increase the oxygen back to normal, the fluorescence goes away. So it's a very nice readout and more sensitive than the previous generation of sensors that were available. Now this is actually another sensor that is based around our Iridium-3, and this probe is actually specifically designed by this group for function in vivo, and so they've conjugated it to a polyvinyl pyrrolidone, and that the function of that is really to enhance the pharmacokinetic properties, so that you can inject and don't have to sort of continuously infuse this compound into animals. Here, the authors are showing, once again, effectiveness in 2D cell culture, as well as 3D cell spheroid models. Now, for hypoxia research, this is actually a very useful tool for an in vitro model, where if cells are grown in little spheres, you end up with the zone in the middle, which is hypoxic, and then you can see that it's delineated by this probe and the red phosphorescence coming from the iridium. And as mentioned, this probe is really engineered to work in vivo, and in this series of images from a non-invasive live imaging experiment in xenograft models, you can see that a single injection is able to detect the hypoxic signaling in this xenograft model for several days. And also, when you excise tissues as well as the tumor, you can see this very specific accumulation within the hypoxic tumor environment. So the important thing about having more sensitive probes is that it allows you to detect hypoxia earlier, and certainly early detection of hypoxia as it relates to cancer signaling is a major goal, because as we all know, it's really helpful to detect things as early as possible to try to intervene with an appropriate therapeutic strategy. So I'd like to think that, the hypoxia research field is advancing with some of these new tools that have been created, some by Thermo Fisher, some by other companies, and that this allows researchers to have new opportunities for studying hypoxia, whether it's in cells or in whole animals. So just in summary, I think it's really important to think about the evolutionary pressures that have, driven the dependence of cells and tissues on atmospheric oxygen, and then all the different kinds of responses mediated by HIF-1 alpha prolyl hydroxylase signaling that drive the adaptive and compensatory responses to hypoxia, some of which are maladaptive and actually exacerbate or contribute to disease pathologies. The central nature, importance of oxygen in human biology is emphasized by this idea that just about every disease state that you look at involves some element of hypoxic signaling, whether it's inflammation, cancer, and I didn't even touch upon neurological disorders, but obviously as the brain is one of the most oxygen-dependent and sensitive organs, understanding hypoxia and its effects on the nervous system is a huge area of interest for medicine. And then also, just to point out that, to get people thinking about how do you study hypoxia in the laboratory, and what is the relative contribution or utility of your 2D cell culture systems. I'm sort of biased. I think that hypoxia is really something that really pertains to in vivo systems in animals, and that you can try to mimic or recreate some of those things in 2D cell culture systems. But ultimately, if we're talking about disease states, then you really need to think about these things in three-dimensional culture models at the least, and probably in whole animals. And then, as I mentioned, there's evolution of new tools, techniques that have been developed to study hypoxia that allow greater sensitivity, control, and hopefully what this means is better and more physiological relevance than what was previously available. And so I encourage you to look at some of the additional resources from Thermo Scientific on our website. If you want to learn more about the EVOS FL Auto and how the OSI system might be able to provide some additional functionality for your research, please visit our website, or you can learn more about the ImageIt hypoxia reagent. There's an application note online that details some of this very nice work that was done here in Eugene to develop this reagent. And then also, molecular tools, TACMan arrays for, being able to sort of query large numbers of RNAs in terms of sensitivity to hypoxia and HIF-related signaling. So with that, I think I'm going to entertain questions, if anyone. Okay, let's see. Looks like we have one from Jet, which is, can hypoxia cause epilepsy, and what would be a mechanism of detecting that? That's a good question. I'm not so, and even though my training is in neuroscience, epilepsy is a complicated phenomenon, disease state, and it probably is involved. But my understanding is that epilepsy is caused by sort of uncontrolled loops of neuronal activity. Now, whether hypoxia sort of exacerbates that kind of signaling, I don't know. I think more generally, hypoxia in the brain is a bigger concern in terms of just leading to neuronal cell death. But I don't know. There might be some literature out there. Once again, there's a lot of really good reviews, some of which I relied upon, to put this presentation together. I encourage you to go to look at that. Okay, another question: will the cell culture medium containing bicarbonate, I see. Do you need to include bicarbonate? And bicarbonate, of course, you want to include in your culture media if your pH is controlled by CO2. So that's more a question about your buffering system. But by and large, most cell culture is done in the presence of 5% CO2. So it probably does need to be in there unless you're working in a heaps buffered system or something like that. Sensitivity of ImageIt hypoxia. Well, as I mentioned, it appears to be more sensitive than some of the previous generation of hypoxia probes. But in terms of the absolute sensitivity, I don't have any numbers to provide to you. Maybe if you, I don't know whether we talk about this in our application note or not, but basically, our application note suggests that you start to see fluorescence from this reagent at 5% environmental oxygen. And once again, I think that there's some sense that is around the concentration where you start to see these adaptive changes. But again, it's very, very cell and tissue dependent. Is the in vivo reagent that I described commercially available yet? That is not a reagent that we are marketing. I would encourage you to reach out to the academic lab that produced it if you were interested in collaborating with them or obtaining some of that reagent. I don't have the reference in front of me, or actually I do. And now this is Shi Chun Jiang's lab at, let's see, it's a collaboration between Nanjing University and Emory in Georgia. So, but that's a Nature Communications paper. I should say that this webinar will be archived online if people want to go back and review slides or have any other more specific questions about things that were covered, but I didn't have time to talk about. Let's see. We're running out of time, so I'm going to try to select here and see. Do you believe that the academic and pharma industry is finally coming around to the idea of culturing and carrying out experiments under low oxygen to better reflect what is observed in vivo? And I'd like to think so. I mean, that's part of the purpose, that I'm giving this presentation is just to, if nothing else, sort of raise awareness and get people thinking about these questions, maybe in a way that they have either taken for granted or hadn't really sort of appreciated as much. But, given that oxygen affects just about everything, and, you look at that list of genes, if you happen to be studying one of those pathways, then you might want to think about it. And certainly the pharma industry, like I said, that there is a lot of energy being placed into hypoxia-targeted therapies, and mainly for cancer, but also for inflammatory diseases as well. And so that's really a reflection of the recognition of the importance of hypoxia signaling, and also the central role that it plays in disease pathology. So I think that my answer would be yes. I think there is an increased awareness and understanding, and let's hope that continues, because it's almost, I liken it to thinking about, like, well, does pH matter? Does temperature matter? I mean, these are sort of all global sort of evolutionary pressures that biology has adapted to. So, you really have to think about all of this when you're looking at your particular piece of biology. Let's see. Would I expect to see major changes in innate immune cell behavior, such as monocytes, if I wanted to look at short-term hypoxia around 6 hours? I guess depending on what your readout is, but my guess is that if you took monocytes and made them very hypoxic for 6 hours, they would probably be eliciting changes that you could measure with any variety of assays. But I think it's very contextual. It depends on exactly what your question is and what your preparation is. But just an easy sort of probably not very satisfying answer would be yes. Monocytes are probably affected by relatively short-term hypoxia. Let's see. Can imaged hypoxia reagent be used in conjunction with chemical hypoxia? Well, once again, chemical induction of hypoxia using, say, DMOG or DFX, that's really like, you're not really inducing hypoxia, you're inducing a hypoxia-like state. So whereas the actual imaged hypoxia reagent is only going to be visible, imageable, so to speak, under conditions of genuine hypoxia. So it sort of goes back to my point that, the chemical tools are useful, but they're really, kind of a poor substitute for actually creating hypoxia, whether it's in vivo or in your cell culture model. So, but in terms of whether we've actually combined that, I don't think we've looked at that. I wouldn't expect those things to interact. And let's see, more questions than I thought. I have problems seeing HIFs on a Western blot. Might it be due to the fact that extracts are prepared in the presence of oxygen? Yeah, I guess that's a fair question. And it goes back to this idea that if you really want to control oxygen across the time course of your entire experiment, well, then everything needs to be done in a controlled environment. So, because oxygen can equilibrate quite rapidly. And so even, if you have a cell culture incubator with a controlled oxygen concentration, the act of opening the door, taking the plate out, moving it to the lab bench to say, change the media or harvest the cells, you're no longer at the same concentration of oxygen that you were in the incubator. Now, whether that is enough to sort of confound your readout is another question, but I think that provided that you're using validated reagents, that are demonstrated to really be specific for HIF-alpha via Western blot, it could be that you may need to consider exactly how your extract is being prepared, or to include some sort of inhibitors in your lysis media. Okay, I think that covers most of the questions. What is the time scale of detection for the nitramidazole probes? I don't know exactly that off the top of my head, but it's a question about a half-life of, how long is the compound clear once it's introduced in the circulation. And I think, a lot of, if you think about what's got to happen for these adducts to form, the nitramidazole probes need to undergo successive rounds of oxidation and reduction. And once they get reduced to a certain terminal state, then they form these covalent adducts. So I think it's on the order of hours, but once again, I'm not sure. There's probably pretty significant literature out there, but that is a limitation of the probes, and why, I wouldn't say they've completely fallen out of favor, but people recognize that they are limited. They're very useful. They're kind of cool molecules, but I think that the emphasis in imaging in general is moving towards doing more things in live cells, live tissues, non-invasive, whole animal imaging. And then if you sort of translate that to patients, then the goal would be to have, very nice non-invasive imaging methods where you could, look very sensitively at patients in terms of understanding whether there are hypoxic environments and detect that early, and maybe you could get can intervene with your therapeutic earlier on in the disease progression. And then also you could potentially monitor the response of the tumors in oncology longitudinally over time using these same reagents. So it goes back to the idea that in vivo imaging is going to be, just more and more widely used in the clinic, and, biopsies are really, yes, they're important, but, we're trying to move away from that basically in the clinical space, and for good reason. So with that, I believe we're out of time. Once again, thank you for joining in and registering. Feel free to reach out to your local Thermo Fisher scientific representative if you have any specific questions. The web is a great source of information. And like I said, feel free to access the archived webinar online after it's posted. And, be happy to follow up with additional questions or try to direct people to the right source for information. So, thanks.